Good afternoon.
Good afternoon, sir.
Shall we start with antibiotics part? Okay. So yesterday we started with the antibiotics chapter, and we studied the introduction part of this class of compounds. And today we'll discuss on penicillin, which comes under the beta-lactam antibiotics. So today we'll understand the chemistry of this class of compounds, specifically the beta-lactams, their numberings, then their mechanism of action, and few structures of under this class. So before starting, we'll briefly go through our previous slides so that we can in continuation with our next part. So in the book-wise definition, yes. रिकॉर्डिंग हो गया आपका? Okay, this is definition given in a book for antibiotics. So antibiotics, as we discussed yesterday, these are the compounds or the chemicals which are released or produced by the microorganisms. The source of all these antibiotics is from microorganisms. They are called as the natural antibiotics. Interesting thing of these chemicals is that they can kill the other microorganisms. Okay, so it is life against the life, or it is against the life, where the one living thing produces chemical and that kills the another living beings. So these antibiotics which are produced from natural sources are natural antibiotics. So there are certain compounds which can be synthesizedly produced and can kill the bacteria. They are the synthetic antibiotics. For example, the sulfonamides and other antibacterial compounds. So these are the highlighted points of the definition. The antibiotics are the compounds which are the product of metabolism of microorganisms. They can be synthetic if we make the derivatives of the natural antibiotics. For example, if there is a structure of penicillin and if you modify the structure of penicillin, then that is also the antibiotic, but it is a semi-synthetic one. Antibiotics are the compounds which can antagonize or inhibit growth and survival of other microorganisms. So this part is very important. They are produced by one microorganisms and killed the another microorganisms. And another important and one of the advantage of antibiotic is that they are active at very low concentration. That is why they are showing the very less side effects or the toxic effects. Because we require very low concentration to kill the microorganisms, or they are highly potent, highly efficient to kill the microorganisms. So this is the basic understanding about the antibiotics. So antibiotics are used in all these bacterial infection. One thing also you should remember that there are certain antibiotics which can kill fungi also. As yesterday we discussed that the antibiotics only kill the bacteria, but that is not the case. Some antibiotics also kill the fungi. Those are called as a antifungal antibiotics. Example is nystatin and griseofulvin. So these two compounds are antifungal antibiotics. They kill the fungi. Well, here in this case, these all the infections right from the brain to the lower organs of our body. Wherever there is a body fluid, then in that body fluid, the microorganisms can grow, multiply, and they can infect the organs which are near to those body fluids. For example, the cerebrospinal fluid, then otitis media, then fluid which is present in the respiratory tract, fluid present in the lungs, GI tract, okay, even in the urinary tract infection. So everywhere the microorganism, wherever they get the nutrients, microorganisms are like the bacteria can grow and they can harm those body organs. Even in the skin also, there is certain sebum and other body fluids are present, and there are also the chances of getting the infection of the bacteria. Obviously, the blood can also be the main area or the region where the bacteria can grow, multiply, and they, through the blood circulation, can infect to the various vital organ of our body. Okay, so wherever the bacterial infection, we can use the antibiotics and can protect our body from this microbial attack. So there are certain antibiotics which are specifically used for the specific diseases. Like we studied in the urinary tract infection, the compounds which are highly water soluble are used. While for the compounds which are used in the CNS system, they should be highly lipophilic in nature, and because they should cross the blood-brain barrier. While the drugs which are used in the respiratory tract infection, they are generally the antibiotics like the erythromycin and all those. Then there are specific drugs for the GI tract infection, likewise. So antibiotics can be classified based on their uses. Antibiotics can also be classified based on their mechanism of action. Okay, so this is also the way we can classify the antibiotics. Like the antibiotics which is act on cell wall, those are beta-lactam, vancomycin, bacitracin, or even polymyxin. These are the antibiotics which can act on a cell wall of bacteria and interrupt the cell wall synthesis and functioning. Okay, so these are the antibiotics under this class. The antibiotics which affecting on the ribosomes like 30S and 50S subunits, these are the tetracycline, aminoglycoside, macrolides, clindamycin, linezolide, chloramphenicol, streptogramins. These all the antibiotics act on the ribosomes. Then the antibiotics which act on the nucleus in the DNA and RNA synthesis, they interfere those nucleotide synthesis. These are the quinolones and rifampin. So rifampin is an anti-tubercular antibiotic, and they act on the RNA polymerase. Okay, so this is also the way we can classify the antibiotics into the different classes. Okay, this is also shown the same information in a tabular form. We can also classify the antibiotics based on their chemical structures. For example, the first class is a beta-lactam antibiotics. The beta-lactam is nothing but a word come from their chemical structure. So there is a lactam ring. Lactam ring is a 4-membered ring. Okay? So like you know that benzene is a 6-membered ring. Then aziridine is a 3-membered ring. Cycloheptane is a 7-membered ring. Similarly, this lactam is a 4-membered ring. Okay? And it having some ketone and functionality also, and that togetherly called as a lactam. There is also the alpha lactam and beta lactam. It is depends upon the stereochemistry of that lactam ring. So this is a chemical name or chemical feature of this class of compound. So based on this chemistry, it is having the name that is beta lactam. Similarly, the aminoglycosides are the nothing but a chemical compounds which is comes under the carbohydrates, which having the glycosides and amino groups which are linked to each other. So they call as a aminoglycosides. Tetracycline is nothing but 4 cyclic ring. 4 cyclic rings. So they call as a tetracyclic or cyclins. So all the compounds having those structural features, they come under this class. Macrolides is a compounds which having a big cyclic structures of maybe 10, 12, 14 or more carbon units. Lincomycin is a particular class of compounds which having a structural feature that we'll see in the next slides. Polypeptides are nothing but a peptide ring. So all of we know that whenever the amino acids link to each other, they call as the peptides. Polypeptides means multiple amino acids or multiple peptides are linked to each other. They called as a polypeptides. So this is how we can classify the antibiotics. Okay. So classification of antibiotics you may see in the books in pharmacology, like based on their chemical, based on their uses. In some books, you also see the classification based on their mechanism of action. Okay, but in chemistry, in medicinal chemistry, when the question comes that write the classification of antibiotics, then you have to write the classification based on their chemistry, okay, their structural features. And now we'll study one by one. We'll start with the beta lactam. Okay, this is the chemical structures just for your understanding that how these different classes looks like. See, the penicillin and cephalosporin are under the beta lactam. So as I told you, beta lactam is nothing but a 4-membered ring. So here I will show you with annotation. So if we can concentrate on cursor, so this is a 4-membered ring and this is called as a beta lactam. So any compound which having this 4-membered ring with a nitrogen and ketone functionality, it is called as a beta lactam, or those compounds under the beta lactam class of compounds. So though there are different compounds like penicillin and cephalosporin, both are having the common feature of beta lactam. So they comes under the one class. Tetracycline, as I told you, there are 4 cyclic rings, 1, 2, 3 and 4. It is called as a tetracycline. Then macrolides, as I told you, it is a big cyclic structure of maybe more than 10, 12 or 14 carbons. So you can see here, so it is not looks like a aromatic ring, as it is having aliphatic features. So it is not a rigid aromatic ring. And also it having the more number of carbons. So it has some bends in their structures, and so it looks like this cyclic structure. And also it having structural feature to bind with the various sugar units. So they are called as a macrolides. So famous example azithromycin, as in last lecture you told about Azi 250, Azi 500. So in that whatever the compound present, that is azithromycin, is looks like this chemically. Okay. Its chemical structure is of macrolide compounds. Lincomycin having these features of multiple hydroxyl units and various heterocycles attached to it. So this is a special class of compound. And as we told that polypeptide. So this is the structure of bacitracin, and bacitracin having these polypeptide units, and these polypeptide units are called as a are under this class. So there are many compounds possible. If you change one amino acid, then there will be different compound. If you change multiple amino acids, again the compounds will change. So you can synthesize or you can isolate the n number of compounds which having the different amino acid linked to each other. Some may be active, some may be inactive. So there is a large scope of research into this area, that which structural features or which amino acids are responsible for activity and which can reduce the activity. Okay. So if you go into the depth of understanding about the polypeptide, it is a lot to learn under this point. Okay. So we'll start with the first one, that is beta-lactam. So as here shown on the slide with arrow, which ring is actually called as a beta-lactam. So in this structure of cephalosporin as well as of penicillin, both are having this 4-membered ring attached to either 5-membered ring or a 6-membered ring. In case of cephalosporin, beta-lactam, which is a 4-member, attached to the 6-membered ring. In case of penicillin, the beta-lactam is attached to the 5-membered ring. Okay. So for this slide, you only understand that beta-lactam is a common in all the compounds under this class. And beta-lactam is nothing but a 4-membered ring with a ketone and nitrogen functional. Penicillin and cephalosporin are the famous drug under this class, but it is not the only two drugs. Okay. There are also the monobactams and carbapenems. This class of compounds are also comes under this category. Okay. But the famous or widely used are the penicillins and the cephalosporins. But there are a number of compounds which are monobactams and number of compounds which are the carbapenem also shows the antibacterial effect. Clear, all of you, about what is meant by the beta-lactam and their examples?
Yes, sir.
Now we'll go in more detail about the chemistry of this compound and then mechanism of action. So this is a common scaffold for penicillin class of the beta-lactam. Okay, so as we only study the penicillin and cephalosporin under this part, because the monobactams and carbapenems will be the too much information for you. Okay, maybe in the next semester we'll add the few points from those headlines, under those headlines, but now we'll only focus on penicillin and cephalosporin. So first we'll see the penicillin under beta-lactam. So in penicillin, as we know that there is a 4-membered ring attached to the 5-membered ring, while in cephalosporin, the 4-membered ring attached to the 6-membered. That is the difference. Now, if we try to numbering this heterocycle, now we call it this as a common scaffold. Okay, so this is a common scaffold, and in this, if we number, then numbering can be start from 1 at nitrogen, or it can be start at 1 at sulfur. So if you read Wilson, what is written there, according to the chemical abstract IUPAC name, the numbering starts from sulfur, but according to the USP, the numbering starts from the nitrogen. Okay, so we have to take either of this to understand the different chemical structures. So according to the book, standard reference book will follow the USP numbering, where the numbering starts from the nitrogen, and it is go like this anti-clockwise. So first number is to the nitrogen, which is in between the 4-membered and 5-membered ring. Then second to this carbon, which is attached to the carboxylic unit. Third to the this carbon which is attached to the dimethyl groups, fourth to the sulfur, fifth again to the carbon which is in between the 4 and 5 membered ring, and then to the sixth to the beta lactam carbon, and 7 to the beta lactam carbonyl group. Okay. So for you all should know this chemical structure or this common scaffold, then only you can number, and if you number well, then you can write the various derivatives and their IUPAC name. Okay. So this is the very basic information, and every one of you should know these things. Okay. So I will repeat very briefly. In a beta lactam, the 4-membered ring attached to the 5-membered ring, where the numbering starts from the nitrogen, and it is goes anti-clockwise. First it is complete the 5-membered and then move toward the 4-membered. So this is how the 1, 2, 3, 4, 5, 6 and 7 numbers are given. So substitutions are generally on the 6th position. So if we check the different penicillins like penicillin G, penicillin V and other penicillins, the substitution takes place at the 6th position, that is at this R group. So in some compound, this R group may be benzene, some compound this R group may be phenoxy, likewise. So based on this R, there are the different types of the penicillins. And each penicillin has different in their mechanism, different in their activity profile. That means some are potent or highly active, some are less active. Because as we change the R group, their water solubility changes, their permeability changes, then their other physicochemical properties like pKa and protein binding, those all the features are changes if you change the R group. Okay? All of you remember that the remaining structure remains intact, it is not changed. Where is the only change? Change is only at the R position. So based on this R position, there are different types of penicillin. Okay, so all of you, okay, if you have notebook, you draw this structure in your notebook so that it will get familiar to you. Draw the structure and also give the numbers. Done?
Yes, sir.
Now can you tell me at which position carboxylic functional group is present?
Second.
At which position amide group is attached? Position at which amide?
Sixth position.
At which position sulfur is present?
Fourth.
So sulfur is also called as a thia, THI. Okay, so it is also called as a thia. And at which position carbonyl group is present? Position of carbonyl group or ketone group?
1.
कार्बोनिल कीटोन 1 पे कार्बोनिल है क्या?
मतलब 6th को अटैच्ड है.
हाँ 6 को अमाइड है. So yes, amide is attached to the 6th position through nitrogen, but at which position there is a carbonyl group directly, the presence of
7th.
At which position the two methyl groups are attached?
Third.
Third. Very good. Now you are well familiar with this structure. Now we'll move to the next part. Okay. So if we cut down this structure, then we'll get the different parts. So these are the parts shown on the slide. The pink color is a beta lactam. Why it is beta? That is shown here. Because it has this particular stereochemistry. So it is called as a beta. If it is having the dot type of the bond, then it is alpha. So beta is towards you. That means this nitrogen is facing towards you, towards the observer. So it is called as a beta. And whenever it is a dot bond, that means that nitrogen is away from the observer. Okay. Now, so this is a beta lactam which is shown in a pink color. That is a one part. Second part of the structure you can see here if you observe the cursor. There it is called as a thia. Thia means sulfur, and aza means nitrogen. So thia aza. Thia aza means there is presence of both thia sulfur and aza nitrogen. And so it is commonly called as a thiazolidine. Thiazolidine ring. So the very close structure to it is the thiazole. Difference between thiazole and thiazolidine is that in case of thiazole, there is a double bonds are present in the structure. There is a presence of double bond. But here there is a no double bond present. So it is called as a thiazolidine. So this is the one part you can see here. So thiazolidine and beta lactams are attached to each other. So this is the, if we see another unit of the structure is called as a carboxylic acid, which is present at the position number 2. Then if you see another functionality is the amide linkage, which is in the blue color and attached at the position number 6. And now R. R is a variable group, and depending upon the R, there are different type of the penicillins that we'll see in the next slides. Now, can you tell me the how many stereo centers or chiral centers in this structure?
3.
3. So this penicillin and its derivatives having the 3 chiral centers. Generally, such questions ask for the objective type of the questions, and for that you should know the details about this. So at which position chiral centers are present?
Position 2.
Yes.
5 and 6.
So at position number 2, 5 and 6 chiral centers are present. So maybe the question asked, in penicillin, the chiral centers are present at the position number 5, 6, 7, 2, 4, 5, 2, 5, 6, 2, 7, 5, like that. Okay, so the right answer is that 2, 5 and 6. At this position, the chiral carbons are present. Okay. This is slide again showing the same thing. Beta lactam, thiazolidine, acyl side chains, like this. Now, this slide is explaining the IUPAC name. Okay. So this slide is explaining the IUPAC name of the benzyl penicillin. Okay. So how we can give the IUPAC name to the compound like penicillin? So now all of you are familiar with the numbering, and also you are familiar with the different functional groups present at the different numbers. So just read the name and see to the structures. Don't bother about the stereochemistry part which is given in the IUPAC name. First see the name of this compound given in the IUPAC form. See, what is it? It is written 3,3-dimethyl. So at position number 3, 3,3-dimethyl. Then 7-oxo. That means at this seventh position, carbonyl is present. So it is called as a 7-oxo. Then see at position number 6. That means this position. At position number 6, there is 2-phenylacetyl. That means this. This is nitrogen is 1 and this is 2. So at position number 2, what is present? Phenylacetyl. 2-phenylacetyl amino means this nitrogen. Okay, so what is at the position number 6? 2-phenylacetyl amino. Then 4-thia. So this sulfur is called as a thia. So 4-thia. 1 means this nitrogen. So now it is this combined ring, that means beta-lactam plus thiazolidine. It is togetherly called as a aza bicycloheptane. So it is togetherly called as a aza. Aza means this nitrogen. Bicyclo means 2 cyclic rings. Heptane. Heptane means 1, 2, 3, 4, 5, 6, 7. 7 atoms are present, so it is called as a heptane. Okay, and at position number 2, carboxylic acid. So this is how the IUPAC name can be designed from the structures and from the numberings. Now you move towards the stereochemistry of this compound. So at position number 2, 5, and 6, these 3 positions are chiral centers, and in case of penicillin, at position number 2 is S configuration, position number 5 is R configuration, and position number 6 is also the R configuration. Okay. So if you remember these 3 positions, 2, 5, and 6, where SRR, that means S at 2, R at 5, and R at 6. Then you can write the stereochemistry also in the IUPAC name. 3,2-heptane means at 1, 2, 3. Okay. I will come back to this point, why it is written as 320. It is explaining that at which part these 2 rings are attached. That means this beta-lactam can be attached to this nitrogen and this carbon, or this carbon and this carbon. So at any position, the beta-lactam can be linked. But 320 is specifically explaining that the beta-lactam is linked to only this carbon and the nitrogen of the thiazolidine ring. But why it is exactly 320, I will explain it in the next lecture. Okay. All of you understand the numberings and the IUPAC name? Any questions?
No, sir.
Now, these are the various examples of penicillin class of compounds, and they are only differ from each other by the R group. Yes, if R change हो रहा है, remaining all the structure is same for all the compounds. Okay, so there are many penicillin compounds because the R can be substituted by the any functionality. But if we change the R with the benzene, it is active. If we change R with the phenoxymethyl, it is also active. If we change R with the dimethoxyphenyl, it is also active. So the number of compounds can be designed, number of compounds can be synthesized, but which are the active, that is important, and which is non-toxic, that is also important. So here are shown the few derivatives of the penicillin which are active and non-toxic and can be used clinically to treat the patients. Okay, this is the common name given. This is the chemical name given. Chemical name explains about the R group, because penicillin is a common word in all the names. What changes? Changes is the this chemical name which explains about the R group in the structure. For example, as just now we seen the structure of benzyl penicillin, it is also called as a penicillin G. So penicillin G is nothing but benzyl penicillin, where the benzyl group is attached to this carbonyl linkage. Okay, carbonyl, after that carbonyl, we have the CH2 phenyl. In case of penicillin V, there is a CH2O phenyl or phenoxymethyl after the carbonyl. So carbonyl, CH2O and phenyl. So this is the structure for penicillin V. Likewise, we can draw 1, 2, 3, 4, 5 and 5, 10. We can draw the 10 penicillin structures if you remember the R groups. Okay, simple. If आपको सिर्फ ये R group ध्यान में रखना है, if you can remember this R group, you can draw the 10 different penicillins because already you have know about the common scaffold of this class of compound. So it is not difficult to draw these structures also. Then if you study well, then you can write the IUPAC name of also all these 10 compounds. That is also not the tough task. And if you write the numbering well, you can easily write the IUPAC name because only you have to remember the change in the R group of the IUPAC name. IUPAC name में जो भी changes है, that will be only at this R position. The remaining IUPAC name also will be the same for all the compounds. Okay, so that is not a difficult task. Okay, so this is about the chemistry of this compound. We also study the structural activity relationship after studying the structures of these compounds in a comparative mode. When we will compare all the structures togetherly, then we will study the structural activity relationship in the next lecture. And now we'll move toward the mechanism of action. Any questions till now?
No, sir.
Now, in our previous slide, we studied that the beta-lactam is a class of compound which act on a cell wall, okay, like the other compounds like polyene antibiotics and other compounds. These compounds are mainly interfere the cell wall synthesis process. Can anybody tell me which other class of compounds we also studied that inhibiting the cell wall synthesis?
Antifungal agents.
Which antifungals? Is it azole class of compound?
Yes, sir.
Can you name few azole?
So ketoconazole, clotrimazole, and fluconazole,
Yes.
oxyconazole.
Okay. Now, if you remember more detail, then can you tell me these azole compounds are inhibiting which biological or biomolecule? Synthesis of which biomolecule?
Ergosterol.
So this ergosterol, we studied that they are specifically present in the fungal cell membrane, while in the human cell membrane or in other bacterial cell membranes, which steroids are present? In fungus, ergosterol is present. The same sterol. Which are the types of sterols present in a human cell membrane? Is it cholesterol?
Yes, sir. Cholesterol and then peptidoglycans.
Yes, very good. So this cholesterol in case of human cell or the ergosterol in case of fungal cell, these are the steroids, steroidal compounds, and if you check the structures of the steroids, They are the rigid and the flat structures. And if you know about the any cell membrane, it consists of the bilayer of the phospholipids. So what are phospholipids? There is a phosphate units at the outer side of the cell membrane and also at the inner side of the cell membrane. Okay. So there is a bilayer, and this bilayer, if we have the figure, we can see. So this is a bilayer where this blue color showing the phospho groups, and inside the phospho groups there is a lipids present. ये सब जो lining दिख रहे हैं, all these linings are the lipids. So any cell membrane, you take up the fungus, bacteria, or the human cell, it is composed of the phospho and lipids. But the problem of this phospholipid is that these are highly flexible in nature. Okay? You just imagine that if agar ye cell membrane sirf phospholipid ka hota, then there will be no rigidity in that structure, because phospholipids are highly flexible. So what is the problem of losing the rigidity? This cell membrane will not have a proper shape. First problem. Second, that it can be break down easily. Okay, because there is no rigid thing in that. It is highly flexible. Even with the pressure of water or the ions, the cell wall can be ruptured, and all the organelles in the cell will come out, or all the toxic substances in the environment will go into the cell. Okay, so that there is a need to have a proper and rigid wall around the cell to protect the organelles present in it. And also it has the function, all of we know the function of cell membrane, it has the function to transport the nutrients and ions from the environment to the cell and from the cell to the environment. Okay. So this is the function of cell membrane, to provide the rigidity, to provide the transport, to provide the shape to the cell, to protect the organelles. These are all the functions of the cell membrane. And these functions of the cell membrane is only because of the rigidity of the cell membrane. And this rigidity is because of the steroids present in the cell wall or the cell membrane. So ergosterol, cholesterol, these are important. And along with these steroids, there are also the important component present called as a polypeptides. Okay. So these polypeptides, you can see in the next slide. Okay. So this is a peptidoglycans. So peptidoglycans are nothing but a polypeptide chains. Okay. So these peptidoglycans and these linear chains of the peptidoglycans are attached to each other by the amino acid chains. Okay. Or they can be they are like a mesh-like structure. So in between the two phospholipids, okay, so in between these two phospholipids, there is a mesh of peptidoglycans. Okay. There is a networking of the peptidoglycans, and these peptidoglycans or the steroids provide the rigidity to the cell membrane, and that helps the cell membrane to have their intact shape, size, and they can protect the cell organelles. Okay, so this is also the same case for the bacterial cells. So these compounds which are acting on the cell membrane, either you take the example of azole antifungal compounds, or you take the example of penicillins or other compounds which are acting on the cell membrane. How these compounds rupture the cell membranes, or how these compounds make the cell membrane fragile? They attack on the steroid synthesis, or they attack on the peptidoglycan synthesis, or peptidoglycan linking with each other. Okay, forming a mesh structure or forming a networking structure. Those properties are interfered by this class of the compounds. So if we read the mechanism of this penicillin, or if we check how these compounds of beta-lactam antibiotics like penicillin, cephalosporin, monobactams, carbapenems, how these compounds act, okay, they first bind to the penicillin binding protein called as PBP. Okay, so penicillin binding protein are nothing but the proteins which help the drug to enter in the cell membrane. Okay, so they're like the transporters here. So after that, when these compounds enter into the cell membrane, they block or inhibit. This word is very important. Cross-linking of peptidoglycan. So as we studied, peptidoglycans form a mesh-like structure between two phospholipids. So this mesh-like structure is very important for a cell membrane to have rigidity. So exactly penicillin interfere in that process of forming the cross-linking, and that is the reason why the cell wall get fragile, leaky, non-intact, and that is the reason why the bacteria is having deficient with the cell wall. It will get completely rupture, burst out, and undergoes lysis or the death of this bacteria. And this is how this beta-lactam shows the bactericidal effect. So this is the mechanism of this class of compound. If you see again, beta-lactam compounds like penicillin, cephalosporin, monobactams, and carbapenems. These compounds bind to the penicillin binding protein. So it is called as a common protein called as penicillin binding protein. They help this drug to enter into the cell membrane, and then this drug inhibit the cross-linking process of the peptidoglycan. That is the reason why the cell membrane get disturbed and deficient, so that the bacteria undergoes lysis and it shows the bactericidal effect. This is the explanation of this mechanism. This is how you can also show the diagrammatically. You can draw the two phospholipid layers, and in between you can show the cross-linking of the various linear chains of the peptidoglycans and how this beta-lactam class of the compounds break down. So here, if you can concentrate on the cursor, here there is a intact link between two layers. Here also there is an intact link between two layers. But wherever it's shown the structure of beta-lactams, you can see here it break down the linkage between the two peptidoglycans. Here also it break down the linkage between two peptidoglycans. So this is how they disturb the networking of these peptidoglycans. Okay, and this is the mechanism. Okay, this is mechanism given in the Wilson. You read it for just a minute. You read it, then we'll go through and we'll stop here. So what is written here? Beta-lactam antibiotics are lethal antibacterials. That means they are bactericidal in nature. They inhibit the bacterial cell wall synthesis that we see. Okay. Now, if you go into the detail of this mechanism, how they inhibit the bacterial cell wall, what is written here? It involves the inhibition of the biosynthesis of the dipeptidoglycan. So, see here, dipeptidoglycan. Dipeptidoglycan means two peptidoglycans are linked to each other. And how they are linked? They are linked by the amino acid linkage. And these compounds are specifically block or interfere the linking process of the peptidoglycans. Okay. And then because of this inhibition of the dipeptidoglycan synthesis, the cell wall loses its strength and rigidity. Also, this penicillin and cephalosporin acetylate a specific bacterial D-transpeptidase enzyme. So D-transpeptidase is the enzyme which actually transfer the peptides or these polypeptides and forms a linkage between the two polypeptide chains. So this enzyme is blocked by this class of the compounds, and hence the further process of linking is getting interfered, thereby rendering it inactive for its role in forming the peptide cross link of two linear peptidoglycan strands by transpeptidation and loss of D-alanine. So D-alanine also having the role in the linking of peptides with each other. So these compounds are actually, if you go in detail, acting on the transpeptidation process. So bacterial D-alanine carboxypeptidase are also inhibited by the beta-lactamase antibody. So there are many propositions of the mechanism that how this drug can inhibit the peptidoglycan linking process. Okay. Understand mechanism of action? Any question?
No, sir.
Okay. Then this slide is for the SAR of this compound. So just now we studied the chemistry. Almost every part of this compound is important. Like amide linkage is important for activity, lactam ring is important, joining of lactam ring to the thiazolidine is important, carboxylic group at position number 2 is important, then stereochemistry at position number 5 and 6 is important. Okay? So almost every feature is important. We'll see in detail their stereochemistry in the next lecture. And as we discuss, so we'll stop here just for your to remind you. We discussed that we'll study the classification of drugs for these 4 chapters: antiviral agents, sulfonamides, antimalarial, and urinary tract infection on Thursday. Hope you are collecting the classifications separately. Okay. So for antivirals, already slides are shared to you. You can just note down the classification of this slide, particularly under the antiviral chapter. For sulfonamides, there are orally absorbable, orally non-absorbable, and mixed sulfonamides. So these slides you can note down in your notebook or separate pages under the classification. Okay. Okay. We'll stop here and we'll discuss tomorrow the SAR of penicillin. Okay.